Okay, everyone, switch on your videos. Let's start today's class.
Ma'am, my Wi-Fi is a little weak today, so I won't be able to turn on my camera.
Okay. So, what is the last thing you people have written in your notebooks? Can you check and tell me?
Ma'am, ROP codes for the protein involved in the replication of plasmid.
All right. So, in the previous class, we had talked about the plasmid, right? All of us now know what plasmid is, right? Now, let us see what are the differences in between the plasmid DNA and the chromosomal DNA. Like, how are they different and why we have given them separate names? Now, when we talk about the plasmid DNA, we know that the plasmid, the chromosomal, both the kinds of DNAs, they are circular. Circular means exactly circular. This. Apart from that, there is a difference. The greatest difference between the plasmid and the chromosomal DNA is that the plasmid DNA is circular. It's circular and it is double stranded. The chromosomal DNA is double stranded, but it can be linear also, like straight, or it can be circular. The plasmid DNA is present without histone proteins, and the chromosomal DNA is present with histone proteins. Histone proteins are present in chromosomal DNA, that is helping in the packaging of the DNA. Now, the plasmid DNA does not carry any kind of vital gene, meaning it is not carrying the gene that is necessary for the organism to stay alive. But the chromosomal DNA, they carry the vital gene. And plasmid DNA, as you people might remember, I said that they are autonomous, meaning they are self-replicated. They can replicate on their own without depending upon the main genome, meaning the chromosomal DNA. They don't depend on that. They don't depend on the, you can say, signals to do the process of replication. They do it on their own. They do not depend on anything else. But the chromosomal DNA, it replicates along with the genome. And this is like the chromosomal DNA is responsible for, you can say, regulating every single thing of the body of the prokaryote. Okay, so the differences between plasmid and chromosomal DNA is clear to you all. Also, did you people have any doubts in the previous topics of this chapter? What about the
No,
other?
ma'am.
Okay. Now we're going to talk about selection using antibiotic resistance. Now you people know that in a plasmid, there are certain sites wherein the restriction sites are present and they code for antibiotic resistance. We saw it is for tetracycline, for ampicillin, right? For different kinds of antibiotics. Now, how can we use that particular resistance as selection basis, right? How can we select those um, plasmids that have been transformed and those that have not been transformed? Like, what is the difference between them and how can we find them out? Now, we know that the ligation of any foreign DNA, it is done at the restriction site. And people might remember that there are so many restriction sites in the E. coli plasmid, like the picture that I showed you. And in that, at one side, there was tetracycline resistance, and at the other side, there was ampicillin resistance. So that will give us like a lot of help in terms of selection of the um, transformant. See, if the ligation of DNA is done at the BAM-H1 restriction site, BAM-H1, right? You people remember that this is a restriction enzyme. And what type of resistance does it provide? Ampicillin or tetracycline?
Tetracycline.
Tetracycline, exactly. So this BAM-H1 at that point of site, tetracycline resistance is present in the vector that we saw, PBR320. Now, when you insert any foreign piece of DNA in this particular site, meaning the restriction site, that is BAM-H1. So now the plasmid will no longer be resistant to tetracycline, right? We discussed it in the previous class, how at whatever point that is giving us the antibiotic resistance, that will be lost in the case of transformant. So due to this, recombinant plasmid will lose the resistance to tetracycline due to insertion of foreign DNA. And we say that this plasmid is now a transformant. It has been transformed because it has now a new gene in it. That is an alien piece of DNA. Alien means foreign, not actually alien. Now the transformant, they can be recognized from the non-transformant by the plating process. Remember I told you that we take a Petri plate, we put the bacteria or the plasmid in it, and then we add ampicillin. Now those, like all of them will grow because none of them have lost the ampicillin resistance. But when we talk about putting tetracycline into the plate, now they will not grow. The ones that have been transformed will not grow. Why? Because of the insertion of foreign DNA, they have lost their tetracycline resistance. So if you add tetracycline to it, they will not be resistant to it anymore and they die. So that is how we find out transformants from non-transformants. So the transformant will grow on the medium containing ampicillin and not grow on the medium containing tetracycline. So now we know which one is transformant and which one is not. Now in this case, antibiotic resistance gene, it helps in selecting the transformants from the non-transformant as one of the gene inactivated due to insertion of alien DNA helps in the selection. But what is the disadvantage of whole entire thing? It's that it is a very tedious process, meaning you have to first put it here, then put it here, then put it here. Like you have to do several plating. And if you have less time and you want the results quicker, this is not ideal, right? Because you have to wait for the observation. So the disadvantage is that the selection of recombinant due to inactivation of antibiotics is a cumbersome procedure because it requires simultaneously plating on two plates having different kinds of antibiotics, ampicillin and tetracycline. So as an alternative, there is another type of selection. This is blue-white selection. You people remember the color forming colonies, the ones that have the alien DNA, they will form the color or not form the color?
Form the color.
Form the color.
That have alien DNA, they will form the color.
No. That has.
See, in this case, normally, you might remember the enzyme name, that is beta-galactosidase, right? So the bacteria that does have the beta-galactosidase, it will make a color on a colony. But if you put any alien DNA into it, now that beta-galactosidase gene will be inhibited or the enzyme will not be synthesized, because of which it will not produce color. So now you can see the color and you can see the colorless cell or the plasmid, and you can easily find out which one is a transformant and which one is not. Now blue-white selection. Okay, so in this type, the selection of recombinant from non-recombinant on the basis of their ability to produce color in the presence of chromogenic substrate, meaning color producing substance. In this case, what will happen? When you put the alien DNA or a recombinant DNA into the vector, then the enzyme beta-galactosidase will sort of get inactivated. And because of this very reason, the inactivation of the gene for synthesis of enzyme, because of the insertion of a foreign DNA into it, will be called as insertional inactivation. This is a term they can ask you, what is insertional inactivation? So you should know that this is what the name only tells us, that because of the insertion of any foreign DNA or an alien piece of DNA causes the inactivation of the genes that codes for enzyme, right? You people are following me? Now the presence of
Yes.
Can you explain again?
See, if a question gets asked as to what is insertional inactivation, right? So insertional inactivation is that phenomenon wherein when we insert an alien or a foreign piece of DNA, and because of that insertion, the genes that are coding for a particular enzyme, any enzyme, Here, beta galactosidase. So this insertion will inactivate that gene, which is responsible for coding for the enzyme. Now, what will happen if the enzyme gets coded? Hmm? What will happen if the enzyme is getting coded?
It will show the color.
It will show the color, it will produce the color. But if the enzyme does not get coded, then it will not show the color, and that is what insertional inactivation is. Is it clear to you all?
Yes, ma'am.
Now, the presence of a chromogenic substrate gives blue colored if the plasmid is a non-recombinant, and it will give white when, meaning it will not, white means it will not produce any color if any foreign gene has been inserted. Any doubts up until now? Okay. Now you people can note down till here, then we'll move forward. Students, I'm just coming in 2 minutes, okay?
Yes, ma'am.
Yes, students, done.
Yes, ma'am.
Yes, ma'am.
Done, ma'am.
Everyone's done.
Yes, ma'am.
Done, ma'am.
Done, students?
Yes,
Yes,
ma'am.
ma'am.
Done, ma'am.
All of you are done? Hmm?
Yes, ma'am.
Done, ma'am.
Done, ma'am.
Done, ma'am.
Done, ma'am.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Yes, ma'am. Done.
All right.
1 minute,
So,
ma'am.
okay.
Done, ma'am.
All right. So up until now, we saw the most common kind of vector, that is the plasmid, and that has been taken out or extracted from the E. coli, right? Escherichia coli. Now let us see what are the some other vectors. See, so apart from plasmid, there are certain organisms, or you can say there are certain creatures or structures which can be used as a vector. Okay, so the first one is bacteriophages. Have you heard about them before? Bacteriophage? What are bacteriophages?
Virus that infect bacteria.
Hmm. The virus that is responsible for causing diseases in bacteria. Those kind of viruses are bacteriophages. So now what happens is that this particular bacteriophage, once it's infect the bacterial cell, it divides so quickly, it divides into thousands and thousands of numbers. So that property that it has, due to which it can divide it like multiple folds, we'll say that is called as the copy number. So the bacteriophages, they have high copy number present. Now, what happens is that we know normally that this will cause disease in the bacteria. But now these bacteriophages, they have been modified in such a way so as to be able to use this to deliver the gene of interest in the case of bacteria. Okay, and it will do the normal function, that is multiplying a lot. Got my point? So this is bacteriophage for being used as a vector in bacteria. Yes.
Ma'am, can you repeat? I didn't understand.
Huh?
I didn't understand.
Oh, yeah, see, just like how plasmid is used to transfer genes into the host organism. Now, apart from plasmid, which is one of the most common type of vector, there are certain other vectors also. One such example of other organism or other structure that you can use as a vector is, the first one is bacteriophages. Normally, what property are we using of bacteriophage? The property that it infects the bacterial cell, meaning it will go inside the bacterial cell and start dividing. And once it does, it divides a lot. So it has a very high copy number. Having a high copy number means it can divide in less time and produce much more number of bacteriophages. Okay. Now, these bacteriophages, they can be used to transfer the gene of interest in bacteria. Because ultimately, if you link the bacteriophage with the gene of interest, whatever you have taken out, if you link it with bacteriophage and you send that bacteriophage into bacteria, so bacteriophage, just like how it has a property of dividing a lot, it will divide. And then the gene of interest will also divide along with it. Clear? Now, vectors that are used for the cloning in plants. Now, these are specific for plants. The one is TI plasmid. You will have to learn more about this in the biotechnology application chapter. There's like a proper descriptive unit about it. But for now, it's very superficial. It's that TI plasmid, the kind of plasmid that is tumor inducing. What is tumor, students? How would you define tumor?
A bunch of cells.
A cluster of cells, right? Very good. So this tumor inducing plasmid, meaning what does it do? When it goes inside the plant, it induces tumor, meaning it forms mass of cell, cluster of cells at several places. Now, this TI plasmid, it has been extracted from a bacteria called as Agrobacterium tumefaciens. This is the name of the bacteria. This bacteria has this plasmid that is known as TI plasmid. Okay. Now, normally, this bacteria that is Agrobacterium tumefaciens, it is a pathogen to plant. Okay, it is a pathogen, meaning once this bacteria gets inside the plant, it causes disease in the plants. And how? Because it has tumor inducing property. It induces tumor in the roots and everything. Okay. So what happens is that we can, since us humans are so intelligent, so we can use this particular plasmid of the bacteria to deliver the gene of interest to the plant cell. Because you people have to understand this one basic thing over here is that no matter whatever is the plasmid, it doesn't matter. The only thing that matters is that the property that they have is the same, meaning their property is to go inside the host and divide. That's what they do. All of the plasmid, no matter what you name it, they'll do that only. So TI plasmid does the same thing. The only thing it does is that it divides a lot and it starts forming a cluster of cell at one place, then another place, then another place. So we can use this TI plasmid like a vector. But you can ask me a question like, ma'am, how can we use it like a vector if it is causing disease in the plant? This will disrupt the whole entire experiment that we are doing. But there are certain techniques to disarm the pathogen. You understand? Disarming. What is the meaning of disarming? Anything?
Um,
Disarming.
like
To let go of something.
To
Let go of something.
to disable or like to stop the
yeah, for
uh,
example,
or
if there's a soldier or if there's a fighter, so you take all of his weapons. You take off his protective coat or whatever he's wearing. So you have disarmed him. So now he'll not be able to fight, isn't it? So same way, if you disarm a bacteria, it means that you take out the pathogenic property from it. So if I take out the pathogenic property from any bacteria, then what property is left with it?
demultiplying properties.
Yeah, the property of division, the property of multiplication. So this is what we do. The TI plasmid. TI means tumor-inducing plasmid of this particular bacteria. We make it, you can say disarm, and then we use it to transfer the piece of interest into the plant cell. The TI plasmid causes diseases in plants, but now it has been disarmed and modified to become a cloning vector because now it is no more pathogenic, and then it is used to deliver the piece of gene of interest into the plant. Clear? Always remember TI plasmid is for plants, not for humans. Now talking about the vectors that are used to transfer the gene, or that are the vectors in the case of animals. The first is retrovirus. Where else have you heard this? Retrovirus. Have you heard it before? You've not heard it before? While I was making the notes for you and I wrote a retrovirus, thing that came to my mind was like, I'm going to ask them, have you heard this before or not? So, yes. And am I only teaching 2 students? Where are the rest of you? Say, have you ever heard about retroviruses before? Yes or no? Have I taught you? Or have I done you a disservice and not taught you?
No,
Hmm. What, ma'am? Yes, ma'am. No,
I
ma'am.
think it sounds familiar.
It sounds familiar.
Yeah,
Okay, so I have taught you.
yes, I think
You think. How many? think ma'am has not taught this and she's asking the question? I'm not talking about vector. I'm just talking about retrovirus. I'm just talking about this term. It doesn't have to be related to this. ऐसे करोगे? ऐसे करोगे तुम लोग? Yeah, okay. If I tell you the chapter, will you be able to tell me?
Yes, sir.
है ना, क्योंकि एग्जाम में तो मैम बगल में बैठी रहेंगी ना. If I'll write something, ma'am will like, no, I'll give you hints, you write it like that. Okay, the chapter is, I don't want to tell you, but okay, I'll tell you. The chapter is Human Health and Disease. Where have you heard retrovirus in Human Health and Disease? अब नहीं बता पाओगे तो फिर बहुत बुरी बात है. What is the silence? Where have you heard about retroviruses? None of you have heard about retrovirus. When I was teaching you AIDS, and I said human immunovirus, right? HIV virus. It is a retrovirus. Now, if I'll ask you why is it a retrovirus, I'm sure no one can tell me that also. Or can you tell me why is it a retrovirus?
Because it's a virus with RNA as its
As
genomic m- as its genomic matter.
genomic matter, genetic material. Yes. So
Genetic material.
the HIV viruses, they are RNA viruses, and any virus that is RNA virus, it is a retrovirus. What property did HIV virus had? What was that special property that it had? See, students, I'm giving you the benefit of the doubt that you are not currently studying that chapter. So you tend to forget things or not be able to answer. But at least tell me the basic things that are very significant about it. It's like very important. What is so special about human health? Students, you like that chapter, I thought. And when you like something, you tend to remember it. None of you remember. You remember that the HIV viruses, they had this particular enzyme called as reverse transcriptase enzyme, because of which from the RNA they could be making the viral DNA. All of you look blank. Anyway, tell me once you're done learning human health and disease, okay? Anyway, so the retrovirus, not just HIV, there are many other viruses also that are RNA viruses. I'll write here so that you will remember that retroviruses are RNA viruses. So these are the viruses that have RNA as their genomic material or genetic material. And what they do is that they cause the normal cells that are there in the case of animals to turn into cancerous cells. Last question. If I say that any normal cell is becoming a cancerous cell, what I mean by that? No, this is very simple question, not very technical. If I say that any cell is cancerous, so I mean that it is
It's harming the body.
it's harming the body. No, don't be so vague. Give me a specific answer. Yes, Mariam, you unmute. So give me the answer.
Okay, can you repeat the question?
The question is, if any cell becomes cancerous, so what is the property of it? I forgot the question, only what I was asking. So I am modifying the question now. Like, what is not so special, but what is detrimental about cancerous cell, or what is so significant about cancerous cells? Like, what is the difference between normal cell and cancer cell? That's the question.
Autoimmune.
Autoimmune? No. What about the rest of the students? And please,
Cancer
those who
cells
have their
have
videos switched on,
carcinogen.
huh?
Cancer cells have carcinogen agents.
No. The normal cells, they become cancerous because of carcinogens, cancer-causing agents. That is different. But I'm asking, what is the difference between a normal cell and a cancerous cell? That is it. And those of you who have your videos switched off, please don't Google and answer. Answer it honestly. What is the difference? None of the questions you people are answering. What is the difference between when I'll tell you the answer, you people will feel very sad. I'm telling you. The answer is very easy. Okay, hint. Hint is that normal cells have something that the cancerous cells lack. Cancerous cells, they have this property of dividing uncontrollably. They divide and divide and divide and divide and divide. So you people, I don't think you remember, but contact inhibition, meaning that is not there in cancerous cells. Anyway, coming back to the topic, these retroviruses, they can be used to transfer the genes into the animal cell, because retroviruses also have a really high copy number. They divide a lot. But again, when we have to send it to the animal cell, we have to disarm it so that it won't cause any disease in the animal cell. Then comes a thing called as shuttle vector. Now this is, I don't think it's there in your NCERT. Just listen. The things that are there in your NCERT, you people are not learning. So I'll not expect you people to learn this. But you should see the shuttle vectors. These are the different kinds of vectors. And they have this, like normally we saw that for plant, there is a different vector. For bacteria, there is a different vector. Shuttle vectors are those vectors that can replicate in both eukaryotes as well as prokaryotes. Okay, normally eukaryotic vectors are different. Prokaryotic vectors are different. But these are the ones that can be replicating in both of them. Just like a shuttlecock. It goes there also, it goes here also. Imagine it like that. Now they contain the two types of origin of replication, one for eukaryotes, one for prokaryotes. They contain selectable markers for eukaryotes and prokaryotes. The example is yeast episomal plasmid, modified PI plasmid. If you want to learn, you can learn. If you don't want to learn, then also it's fine. It's not going to come in the exam. Okay, because it's not there in the NCERT. Any doubts up until now? Sure. Anything you want to ask me?
No, ma'am.
Shuttle vector is there in NCERT.
I don't think so. Check it once.
Okay.
I don't think so. It's there. But this is just for your knowledge. And one more thing, students, you people are enjoying this chapter, right? Huh? It's not boring.
It's not boring, ma'am, but it's a lot of complicated terms.
Because this is something you people have never studied.
It's hard.
It's hard. It's hard.
A bit.
Okay, see, I sort of figured that it will be hard because of a lot of technicalities that this chapter has. Right? There are many new terms that you people have never heard about. So what I was thinking that once we are done or through with the chapter, we'll do a revision also, a proper revision, wherein I'll teach this chapter yet again, not in depth, just like how I teach you right now, but we'll go through the chapter yet again so as to make things clear. And when you'll study the application of biotechnology, then you will see that here we are understanding what are the stuff that we are doing to make a recombinant DNA. Like what are the practices that we are doing in the lab. And then we're going to see how are we using it in the real world. Okay. So just keep the interest. I understand that you can find it hard. I'll make it easy for you. Don't worry. All right. Now, first you note down till here, then we'll move forward. We'll go little by little, okay?
Done, ma'am.
All of you are done?
Yes, ma'am.
Done, ma'am.
Done, ma'am.
Done, ma'am.
Done, ma'am.